Now we are going to understand the oogenesis, means formation of female gamete. Here we are discussing about human, right? So there is a difference between spermatogenesis and oogenesis. So one by one, try to understand. The differentiation and maturation of ova differ from spermatogenesis in several ways. First, one, while the cells from which sperm develop continue to divide by mitosis throughout the male reproductive period, but this is not in the case of female. In female, what is happening? In human, thousands of oogonia rapidly divide between second to 7 months of gestation and produce 7 millions of oogonia. However, after the 7 months, the number of germ cells drop drastically. Many oogonia die, while remaining oogonia enter into prophet 1 of meiosis 1 and get arrested at diplotin stage until puberty. Right? उसमें हम डिप्लोटिन बोल रहे थे, शायद गलत बोल गए थे, राइट? मियोसिस 1 में ही स्टॉप होता है, राइट? क्लियर और नॉट क्लियर? ओके, बिटवीन बर्थ एंड puberty the egg cell enlarges and follicle around them grow. At puberty, follicle stimulating hormone stimulate a follicle to resume meiosis and it is formed secondary oocyte and a polar body with less cytoplasm. Thus here we see a case of unequal cytokinesis. Meiosis then again stop at metaphase 2 of meiosis 2, which remains inactivated unless reactivated by the sperm. These are the steps for the oogenesis process. So see, read what is happening. There is a formation of primary oocyte, then primary follicle around the primary oocyte, and then there developing follicle, then formation of secondary oocyte. Before that, there is formation of one polar body, and then it will go for the ovulation process, and after that, there is a formation of ovum and one polar body. In this way, 3 polar body and one ovum formation takes place. See, this point is important to understand. What is happening? Before birth, oogonia form generally here. So see here that oogonia is diploid, right? Diploid, right? So by the first mitotic division, there is a formation of primary oocyte and arrested until the puberty, right? This is at prophet 1 at diplotin stage. Then what is happening there? Then, by action of some triggering factor, I will explain. By getting some triggering factor, this what is happening there? First meiosis completion. Here, one polar, that is unequal cytokinesis. One polar body and this is secondary oocyte. Secondary oocyte. So again, what is happening that secondary oocyte, it will remain stable at metaphase 2 stage, right? And whenever there is puberty, then FSH starts beginning to produce what? Here, secondary oocyte, right? There is ovum release, right? And then what happened that this is not completed meiosis, right? When there is entry of sperm, then this division, meiotic 2 division complete, and there is formation of second polar body, and there is that is egg for fertilization. So here that completion of first meiosis, FSH is required, and completion of second meiosis, that sperm is required. So see here, same thing here, what is happening that what primordial germ cell, then there will continuously mitotic division, and that will first primary oocyte formation present at birth, arrested In prophet of meiosis 1, what will happen that after getting FSH, there is a completion of first polar body, that is unequal cytokinesis, and then here there is secondary oocyte, and arrested metaphase of meiosis 2. Right? Metaphase of meiosis 2. Then the process of ovulation, there is a secretion of ovum, and then the formation takes place. Then second polar body, and there is fertilization. Same thing, you see that. Oogonia, primary oocyte, meiosis 1 completed, secondary oocyte, primary oocyte, see here. Primary oocyte, there is meiosis 2 completed, then see that secondary oocyte, and then see that what is happening finally. 1 ovum and 3 polar body. Right? So diagram is clear. See, same thing, that is oogonia, primary oocyte, arrested at prophet 1, present at birth, and then what is happening that? Here, see here. Primary oocyte arrested prophet 1 at birth, present at birth, so primordial follicle. Then you can see that there is a primordial follicle growing, and this is known as Graafian follicle. At the process of ovulation, what happens that this Graafian follicle will form corpus luteum, and here this release is not over. This is secondary oocyte. So secondary oocyte, by the process of ovulation, when the sperm release takes place, you see then formation of ovum, that is fertilized egg, and then polar body. And here one, that is again two polar body. So three polar body and one fertilized egg. Clear? This is the structure of egg, mature egg. Important to understand that where is plasma membrane, then first layer, that layer of follicular cell, that is corona radiata, that is second layer. Layer of gel composed of glycoprotein, zona pellucida, then cortical granules. So see that first plasma membrane, then first layer, that is zona pellucida, then second layer, that is corona radiata, and here see cortical granules. These granules also play vital role to stop polyesperm. You will study later on. So oogenesis vary among species to species. Like sea urchin and frog routinely produce hundred or thousand of eggs at a time. Human and most mammals, only a few eggs are produced during the lifetime of an individual, even during oogenesis in amphibia. Why I am trying to tell about amphibia? That in human there is internal fertilization, and in amphibia there is external fertilization. So we will study about amphibia. So in amphibia, what is happening? That two phases. One is multiplication phase, and second one is growth phase. So multiplication phase, what is happening? That primordial germ cell, that is mitosis, oogonia, then mitosis, then the formation of oocyte. So no conspicuous changes has been traced up to the formation of primary oocyte, right? And growth phase, what is happening in oogenesis, which had long until primary oocyte doesn't complete meiosis 1, can be divided into two phases. That is pre-vitalogenesis, and then another one is vitalogenesis. So pre-vitalogenesis complete before diploidic stage, that is cytoplasmic and nuclear material grow, no yolk synthesis, and vitalogenesis start at diploidic stage, synthesis of yolk. So see that what is happening in pre-vitalogenesis. Pre-vitalogenesis में mRNA, tRNA, RNA न्यूक्लियस साइज इंक्रीज कर गया, न्यूक्लियोइड साइज इंक्रीज कर गया, माइटोकॉन्ड्रिया इंक्रीज कर गया, कोर्टिकल ग्रैन्यूल्स जो है और फोलिकुलर सेल और नर्व सेल्स जो वो अवेलेबल हो गया, ठीक है. But in vitalogenesis, what is happening that there is a formation of yolk. Pre-vitalogenesis, one step. Vitalogenesis, another step. Why this is happening? Right? Why this is happening? So changes during pre-vitalogenesis. Production of large amount of nucleus sac. Nucleus of the oocyte also enlarges. Due to large amount of nucleus mass, the nucleus seems to be blotted with fluid called germinal vesicle. So see that the egg is going for the process of production. So there is a need of more amount of these things, right? So during the developing stage, certain chromosomes stretch out large loops of DNA, causing the chromosome to resemble a lamp growth. These lamp growth chromosomes can be revealed at the site of RNA synthesis by in situ hybridization. See here, that's a figure of diploid chromosome of newt Criteria testaceous after incubation with radioactive stone mRNA. It is obvious that it is located on the loop of Lambus chromosome. What is the purpose of Lambus? बताया था, कोई बताओ? Lambus का purpose क्या है? वो खुल जाता है डीएनए का अमाउंट नहीं बढ़ता, खुल जाता है और जिससे मोर mRNA सिंथेसिस और मोर प्रोटीन सिंथेसिस होता है बिकॉज आगे डेवलपमेंट होना है, राइट? तो देखो नेचर ने कितना खूबसूरत बनाया। लैम्ब्रोस क्रोमोसोम इज इंपॉर्टेंट इन द प्री वाइटोलोजेनेसिस स्टेज। Vitalogenesis stage, liver produce inactive precursor vitalogenin, transported through blood through ovary enzyme protein kinase, convert it into active vitalogenin mitochondria. See that what happens there? Here gonadotropin release, hormone release, okay? Then from follicles there is formation, there is secretion of estrogen. You see that there is estrogen. Liver, there is vitalogenesis, formation of vitalogenin, and there is a vitalogenin intake, reuptake हो गया, ये भीतर चला गया, right? So this is known as vitalogenesis, right? तो vitalogenin जो है वो भीतर है। अब see that एनिमल पोल और वेजिटल पोल कैसे डिसाइड करते हैं, राइट? एनिमल पोल और वेजिटल पोल कैसे डिसाइड करते हैं? See, it is during oogenesis that animal and vegetal poles of an egg are specified, right? Vitalogenesis uptake in uniform around the surface of oocyte, however, its movement within the oocyte differs. When young platelets are formed in future animal hemisphere, they start to move toward the center of cell. However, vegetal hemisphere young platelets do not move actively. As a result of this, the amount of yolk ingredient in the vegetal hemisphere, the mechanism of such differentiation translocation is still unknown. So what is happening that in egg? What is happening that यहाँ से जो vitalogenin throughout reuptake हुआ है, लेकिन इस साइड में वाइटोजेनिन का मूवमेंट होता है सेंटर की तरफ, इस साइड में मूवमेंट नहीं होता है उतना फास्ट या बहुत स्लो होता है, जिसके वजह से सेंटर साइड जो है वो थोड़ा सा कम योगी हो जाता है, राइट? ये इस साइड जिसको कि एनिमल पोल बोलते हैं, एंड इस साइड ज्यादा योगी हो जाता है, उसको वेजिटल पोल बोलते हैं। Animal and vegetable pole were specified हो जाता है। Thus, during vitalogenesis is not only yolk which is distributed differentially, but organelles are also distributed distinctly in future animal and vegetal hemisphere. As vitalogenesis approaches to the end, cortical granules, mitochondria, and pigment granules arrange themselves around the periphery of oocyte cytoplasm. And see, what is germinal vesicle breakup? ये कल आप लोगों को क्लास में बताया था, very important, खूब क्वेश्चन इससे पूछता है। So after vitalogenesis at different stage, amphibian primary oocyte wait for the progesterone signal. Thus, resumption of meiosis one in frogs primary oocyte require progesterone, which is secreted from follicle cell. Within 6 hours of release of progesterone, germinal vesicle breakdown occur, and soon after the meiotic division one, mature ovary release over by process called ovulation. However, the ovulated egg is still in second meiotic stage at metaphase 2. यहाँ देखिए, यहाँ interphase stage है यहाँ, right? अब ये first meiotic metaphase stage भी है, यहाँ MPF, माइटोसिस प्रोमोटिंग फैक्टर है, MPF है, ये एक्टिव है, राइट? एक्टिव है, राइट? जब ये एक्टिव जब हो जाएगा, जब ये एक्टिव, देखो यहाँ ये जो है एक्टिव अगर नहीं रहेगा, फॉस्फोराइलेटेड नहीं रहेगा तो एक्टिव नहीं रहेगा, और जब ये एक्टिव नहीं रहेगा तो हॉल्ट हो जाता है, और जैसे ही एक्टिव हो जाता है, बिकॉज प्रोजेस्टेरोन आया, सीमॉस ने इसको फॉस्फोराइलेट कर दिया, तो ये जो एमपीएफ साइकिल P34 था, P34 फॉस्फोराइलेट हुआ, ये एक्टिव हो गए। और जैसे ही एक्टिव होता है, फर्स्ट मेयोटिक स्टेज जो है कंप्लीट होता है, अब प्रोफेज 1 से जो है, प्रोफेज 1 से मेटाफेज 1 में जो है मूव कर जाता है, राइट। तो सी हियर दैट फर्स्ट मेयोटिक मेटाफेज, और फिर जो है ये जो है कंप्लीट होने के बाद सेकेंड मेयोटिक में एक देखो यहाँ एक कवर सेल बन गया, और एक जो है यहाँ सेकेंडरी oocyte बन गया, राइट। अब ये सेकेंडरी oocyte सेकेंड मेयोटिक मेटाफेज में जो है स्टॉप हो जाता है। क्यों स्टॉप हो जाता है? क्योंकि CSF है, CSF जो है वो CSF है, CSF इसके बैरियर का काम करता है, साइटोस्टैटिक फैक्टर इसका बैरियर का काम करता है। लेकिन जैसे ही फर्टिलाइजेशन हुआ, स्पर्म का एंट्री हुआ, कैल्शियम का इन्फ्लक्स हुआ, कैल्शियम जो है कलमोडलिन प्रोटीन को एक्टिव किया, कैल्शियम PK2 एंजाइम और कैल्पेन को एक्टिव किया, और ये दोनों कैल्पेन और PK2 ये CSF को डिग्रेड कर देता है, इनएक्टिव कर देता है, जिसके वजह से ये जो बैरियर था वो खत्म हो गया। और नाउ ये जो है सेकंड मेयोटिक से अगेन डिवीजन हुआ, और जो है एक जो है यहाँ पर जो है देखो कंप्लीट फर्टिलाइज होगा, कंप्लीट फर्टिलाइज सेल एंड 3 व्हाट पोलर सेल्स का फॉर्मेशन, राइट। अब ये बहुत इंपॉर्टेंट है, कल लिखाया था तुम लोगों को कि मोस्ट स्पेसिफिक ऑफ एनिमल ग्रुप स्पर्म रिटर्न्स बिफोर द egg nucleus had completed symbiosis। प्राइमरी कब इंटर करता है? प्राइमरी oocyte स्टेज में, राउंड वॉर्म में, ग्लाउंड में, फ्रॉक्स में और बर्ड्स में, एट फर्स्ट मेटा स्टेज में, इंसेक्ट्स में एंड स्टारफिश में। हम लोगों में एट सेकंड मेटाफेज स्टेज में ह्यूमन, मोस्ट ऑफ द एम्फीबियन, फिशेस एंड ब्रैंचियोस्टोमा, एंड कंप्लीट मेयोसिस सी एनिमल में। ये कई जगह क्वेश्चन पूछे जाते हैं, और ये जो है तुमको टाइप ऑफ मेम्ब्रेन ऑफ एक ऑफ डिफरेंट स्पीशीज, ये भी जानना जरूरी है, राइट। There are 3 types of membrane in different species: primary membrane, secondary membrane, and tertiary membrane, right? Naming of different types of membrane vary from a species to species are given below। देखो प्राइमरी मेम्ब्रेन को वाइटलाइट मेम्ब्रेन कहते हैं, किसमें? इंसेक्ट्स में, एम्फीबियन में, मोलस्क में, बर्ड्स में, सी में। प्राइमरी मेम्ब्रेन का दूसरा नाम जोना रेडियाटा भी है, बहुत कंफ्यूज बच्चे करते हैं पेलिसेडा रेडियाटा में। तो जोना रेडियाटा है जो कि फिश, कार्टिलेजिनस फिश में पाया जाता है, और जोना पेलिसेडा जो है वो भी प्राइमरी मेम्ब्रेन है, किसमें? मैमल्स। देखो तो ये प्राइमरी मेम्ब्रेन हुआ। प्राइमरी मेम्ब्रेन ऑलवेज हेल्प इन स्पर्म एग रिप्रोडक्शन। दे आर मेड अप ऑफ ग्लाइकोपोलिसैकेराइड व्हिच later converted into glycosaccharide। देन सेकेंडरी मेम्ब्रेन, अब देखो जोना रेडियाटा, कोरोना रेडियाटा में कंफ्यूज मत करना। कोरोना रेडियाटा सेकेंडरी मेम्ब्रेन होती है, जो कि मैमल में, राइट। और सच मेम्ब्रेन नॉट फाउंड इन रेप्टाइल एंड बर्ड बॉडी में, ये सेकेंडरी मेम्ब्रेन नहीं पाया जाता। टर्सरी मेम्ब्रेन, these membranes are in different animal group but absent in mammal except prototheria, prototheria and platypus में पाया जाता है। These are references, and please don't use this slide for the research purposes, only for study purposes, right?